Good evening, everyone. I begin by acknowledging the traditional owners of the land we are on this evening, the Wurundjeri Woiwurrung people. I also acknowledge the traditional owners of the lands on which our online audience is joining us tonight. And I appreciate that Aboriginal and Torres Strait Islander people are Australia's first scientists with deep and enduring knowledge of the land, waters and skies. I pay my respects to elders past and present, and I welcome all First Nations people joining us here this evening. I make this acknowledgement, and in doing so, we commit to respectful and responsible conduct towards all others according to the traditional laws of this land. As the Dean of Science, it is my great pleasure that I welcome you to the final of the Big Science Pitch for 2024. This competition for early career academics originated in the School of Biosciences in 2021. This year, for the first time, additional faculty funding has meant that we could open it up to early career academics from all of our schools. And I'm thrilled to have 5 different schools represented here in the final. And this is a significant opportunity. Our 6 finalists have each already won $10,000 by impressing the first round judges with their succinct and persuasive research proposals. Tonight, our finalists are competing for an additional $50,000 in research funding. They have been given 3 minutes to pitch their research idea using clear language and compelling argument to persuade our panel of judges and our public audience that their proposal has the highest potential value in terms of positive social, economic, scientific and environmental impact. Our judging panel will award $15,000 for first place, $10,000 for second place and $5,000 for third place. 3 of our finalists, those from the School of Biosciences and the Melbourne Veterinary School, are also competing for the Tim Winton's Choice Award of $10,000 funded by the Australian Native Animals Trust. In addition, you, the public, will vote for the People's Choice Award, which is an additional $10,000. But beyond these cash prizes, participation in the Big Science Pitch brings other benefits. And I would like to quote one of last year's finalists, Wing Yan Chan. She said, "Participating in the Big Science Pitch really helped me polish my science communication and interview skills. Those skills made a big difference when I was in the final round of interviews for the Westpac Research Fellowship and helped me to win $465,000 in fellowships." It is a compelling example of how building your communication capabilities can open new opportunities which can be career defining. As Dean of this great faculty, I am passionate about investing in training, support, and opportunities and guidance to uplift our staff and students. I am especially keen to nurture our early career academics who will carry forward our faculty's legacy as in turn they become the leaders. The Big Science Pitch is an investment in the capabilities, connections and confidence of our emerging leaders. I admire all of our competitors for being brave enough to submit their pitches. I trust that all entrants, regardless whether they made the final or not, found it a beneficial experience that will help to define their research proposals and hone their presentation skills. I encourage you all to learn from tonight's outcomes and try again next year. The Big Science Pitch is also a wonderful public opportunity to showcase a variety of research from across the faculty and, importantly, to spotlight some of our remarkable people. And now I'd like to introduce your mistress of ceremonies for this event, the marvellous Associate Professor Jen Martin. Jen works as a field ecologist before founding the University of Melbourne Science Communication Teaching Program, which teaches scientists of all disciplines how to engage and effectively communicate. She also practices what she preaches. She's been talking about science weekly on 3RRR, Australia's largest community radio station, for nearly 20 years. And she is the host of the university's podcast, Let's Talk SciComm. In her spare time, Jen writes for a variety of publications and was named the 2019 Unsung Hero of Science, Australian Science Communication. Please welcome Jen.
Thank you so much, Moira, and good evening and welcome to one of my very favourite events on both the university calendar and the science calendar. You are in for a real treat tonight with the big science pitch. And as you've just heard, I believe that it is so important for scientists to be able to communicate their work in effective and engaging ways to different audiences. And tonight we are going to witness some absolutely wonderful science communication, I know. So this is our 4th annual Big Science Pitch, and I'm sure some of you are joining us for the 2nd or 3rd or 4th time. Welcome back. We are so glad you've decided to join us again this evening. But as you've heard, it is very exciting this year that for the first time we have extended the Big Science Pitch to invite early career academics from all schools in the Faculty of Science to join, which means I'm sure some of you are here for the very first time, and welcome to you. So I would love to know in the chat where you are joining us from. I'll do some shout outs in a moment. I know we have a wonderful international group of finalists this evening, so I'm sure we have friends and families from all over the world joining us. Oh, and I can see we have some people joining us. So we've got Munich, a place that I adore and have spent lots of time in. We have people from Wales, from South Morang, from Rome. Welcome from Rome. Ballarat, Townsville, Perth, Armidale, Indonesia, London. This is an extravaganza of international guests. This is wonderful. I'll wait for a few more to come in, but I will just reiterate what Moira said. We do have an esteemed judging panel joining us this evening, and you'll get to meet them a little later. I will introduce you to them. And I'm very excited that we are going to be awarding, there you are, you can see them. I'm very excited that we are going to be awarding the Tim Winton Award this evening. Tim is patron of our Native Australian Animals Trust and managed to take time amidst his very busy book tour for his extraordinary new book, Juice. If you haven't read it yet, I can highly recommend it. And he has chosen his favourite pitch, so I will share that with you later. But in addition to the prizes that our judges and Tim Winton will be awarding, you have a very important job. It is your job to decide on the People's Choice Award. It is all up to you. So you will get to cast your vote for the People's Choice Award once we've heard from all of our pitches. So please just keep in mind as we go along who it is that you might want to vote for. And keep your eye on the chat for the link to the People's Choice voting links and various other updates through the night. But the other way we'd like to get you involved, or for you to become involved, is you need to let me know what questions you would like to ask our pitchers. So each pitcher will have 3 minutes to share their research with you, and then we'll have about 5 minutes for me to pose questions to them that you have given to me. So please make sure as soon as you think of something, don't wait till the end of the pitch, as soon as you think of something you'd like me to ask, put it into the Q&A box so I can ask on your behalf, and we'll get through as many questions as we can. And thank you again for letting us know where you're from. We also have someone from the Philippines and South Australia. So welcome to everybody who's here. So it is time to get started. And our first pitcher tonight is Dr. Rebecca Webb. Rebecca is a microbiologist with a lifelong obsession with frogs, and she's from the Melbourne Veterinary School. In her work, she's developing tools to understand and combat amphibian diseases. Now, Beck also plays roller derby, and she's told me that her roller derby alter ego is toxic bacteria. We heard what you did there, Beck. Over to Rebecca for our first pitch of the evening.
How many corroboree frogs can you fit on one bench? Now, this might sound like an opening line from a joke, but it's an actual dilemma we face in amphibian conservation. Although, if you ask me how many corroboree frogs you might see in the wild, the answer is unfortunately not a lot, because they've been almost completely wiped out by an introduced disease called chytrid fungus. And they're not alone. Chytrid has decimated hundreds of amphibian species worldwide. It's my dream to see the corroboree frogs back out in the wild where they belong, but the chytrid fungus is still out there. So before we can release any of our captive bred animals, we need to better understand this disease. How does the chytrid fungus infect and kill frogs? What factors make it such an effective pathogen? And what can we do to help increase frog disease resistance? Now, I could experimentally infect some of our precious corroboree frogs with chytrid in the lab, but this is expensive, time-consuming, and let's be honest, it's going to take up a lot of my bench space. But what if I scaled this down and instead used lab-grown frog cells? This is cheaper, faster, and the best part is I can fit the equivalent of 96 different frogs in space the size of a block of chocolate. Now that's going to free up some serious bench space. But all jokes aside, this is not about saving bench space. This is about saving species. This is about finding ways to help endangered frogs without having to experiment on the animals themselves, especially when there are so few left. Another advantage of using this scaled down cell model is it means I can use gene editing to precisely manipulate the interaction between frog cell and fungus, creating a customizable tool to tease apart exactly which factors make this disease so deadly. Gene editing allows me to control the expression of individual genes so I can see how important they are. As a start, I've edited a gene that makes the chytrid glow bright red, so I can actually watch the chytrid infect and kill cells in real time. Now I know it can work, I will change other traits to discover whether they make the chytrid better or worse at causing disease. But there are always two sides to every infection, and the great thing about this idea is not only can I look at the fungus, but I can also control gene expression in the frog cells as well. This will be a game changer for my colleagues who are working hard to selectively breed corroboree frogs that are more disease resistant, because I can tell them which traits to encourage and which to avoid. So if you like the idea of this efficient, cost-effective and ethical conservation tool, please vote for me. You'll be helping to find ways to make the chytrid fungus less deadly and our frogs more resistant, which is an important first step towards getting the corroboree frogs out of the lab and back into the wild. And just imagine how much bench space that will save me. Thank you.
Now, you'll notice that Rebecca actually isn't in the studio with us, and that's because she's actually in the US right now, presenting her work at the Amphibian Disease Conference in Nashville, which she tells me is a fantastic collaborative group of researchers who are working together to find solutions to save frogs. And Becca's got up literally in the middle of the night to join us live. So, hey, Beck, how are you feeling? Sleepy?
Yeah, a little bit. How are you?
Congratulations on your amazing pitch. And we've got some fantastic questions here for you. The first question I've got for you is, are there any frog species who are resistant to this fungus pathogen that we can learn from for disease resistance mechanisms?
That is an excellent question, and they are. And in the example I gave of the corroboree frog, there's actually a close cousin of the corroboree frog, the common froglet, which lives in the same environment, experiencing the same amount of chytrid exposure, but doesn't seem to die from the disease. So some colleagues of mine are actually sequencing the genomes of the corroboree frog and comparing it to the common froglet to see if we can learn some of the secrets about why one species is so susceptible and the other one might be resistant.
Fantastic, Beck. That's clearly an excellent question and pretty exciting outcomes there. The next question I've got for you is, what factors would you target to help influence virulence in the chytrid fungus?
Another very good question. I mean, that's what I want to design this model to find out. So there are a few candidates, genes in the fungus that other researchers have determined might be involved in virulence, because they're upregulated when the fungus is infecting frogs versus when it's not. And also they've looked at some close non-pathogenic relatives of the fungus, and they don't have these genes. So that's given us a couple of candidate genes that we will start off with targeting.
Yeah, brilliant. And how awesome is our audience asking you excellent questions.
Questions.
The next question I have for you is, can you give some examples of the genes that might be altered in corroboree frogs?
That is also a good question. That one's a little bit outside of my field of expertise, but that's why I want to set up this model so I can then provide this resource for other people who know more about frog immunity. So we can collaborate and they can give me which genes they want to look at.
Yep. Yeah, that makes sense. And someone else has asked whether this approach is going to have implications for other frogs / fungi. So I guess whether there's other frog-fungi interactions that we need to know about.
I definitely think it will help other frogs for sure. So as a start, I want to start with just a commercially available frog cell line. So that will be applicable to all frogs. And then once I've got that going, move on to a more specialised corboree frog and some of our other endangered Australian frogs. And in terms of the second part of the question, there's a salamander chytrid fungus.
Oh, really? I didn't know that.
Yeah. That's not such a problem for us in Australia because we don't have salamanders, but definitely a problem elsewhere in the world. And there's also other amphibian diseases such as rhinovirus. So this system will be able to, once developed, we'll be able to test a few different diseases. So that's why I think it's such a good idea.
Yeah, super important work. Wonderful question on a slightly different tack, but one I can't wait to hear your answer to is, please tell us, why are frogs important to our ecosystem?
Oh, well, I've always loved frogs. I mean, there's a lot of them. They play an important role. Tadpoles eat detritus in the streams. Adult frogs eat mosquitoes. But also, how can you not love frogs? They're just the greatest.
As a lifelong frog fan, I'm not going to argue with you for a moment. I think we've got time for one more question, and that is, can you tell us roughly how many corroboree frogs there still are in the wild, and do we know that?
Oh, again, not exactly my area of expertise, but basically very few. I would say less than 100. There are two species of corroboree frogs. So there's the northern and the southern. The northerns are doing ever so slightly better than the southern species, but still both of them critically endangered and pretty much reliant on captive husbandry to keep the species going.
Which both makes me incredibly sad, but incredibly grateful that people like you and other amazing researchers are working so hard and so innovatively to come up with ways to protect our native frogs. So thank you, Beck. Thank you for the wonderful work you're doing, and thank you for getting up in the middle of the night to be with us. I wish I could say go and have a nice snooze, but I'm not going to say that because you need to stick around for the rest of the event. Maybe you can just have a little cat nap for a few minutes, but we will see you later on after we hear the other pictures. So thank you, Beck. That was wonderful.
Thank you.
All right. It is now time to welcome our second pitcher to the stage, who is Dr. Marie Dade. And Marie is a member of the School of Agriculture, Food and Ecosystem Sciences, and she studies the environment, health and well-being benefits that urban green spaces provide, and how the design and management of green spaces impact these benefits. And part of Marie's passion for green spaces is her love of native Australian orchids. And she tells me that every spring she likes to go wandering through local bush reserves looking for native orchids, which sounds delightful to me. So over to you, Marie.
I want you to think about the last time you were outside in nature. Maybe it was a park with lots of big trees, or a garden outside your office with lots of brightly coloured flowers. Or perhaps it was just a small patch of grass where you could hear the birds singing. Why did you go to that place, and how did you feel after you visited? All of these places that you visited are examples of what we call green spaces, which include all the different types of natural areas we visit. And if I was to collect all your responses to my questions, I would likely notice two things. Firstly, that most of you visited that green space to relax, exercise, or improve your well-being in some way. These well-being benefits we receive from green spaces are called ecosystem services. Green spaces also provide other more tangible ecosystem services such as food and climate regulation, as well as providing important space for plants and animals. These ecosystem services that green spaces provide are important to our well-being. The second thing I would likely notice in your responses is that the last green space you visited was probably quite small in size and located in a city. A big number of people live in cities. All these people need access to green space to receive the ecosystem services that support their well-being. The more green space in a city, the more sustainable that city is likely to be. But finding space to add greenery in cities is a huge challenge. There's a lot of competition for space in cities. Urban planners have to get creative, finding little pockets of space in our cities that can be repurposed into small green patches. The size of these small green spaces could range from the size of your bedroom up to the size of a small suburban house. While these small green spaces in cities are an increasingly common sight, we don't know much about who is using them and what ecosystem services are being provided. While we know a lot about ecosystem services that other green spaces in cities provide, like big traditional parks, these small green spaces are very different in design and style, so they could be potentially providing really different benefits. My research aims to better understand the ecosystem services that small green spaces in cities are providing. I plan to survey Melbourne residents to determine the ecosystem services they receive from small green spaces, and also determine who is benefiting the most from these green spaces. To do this, I will ask survey questions such as, which small green spaces have you visited recently? What activities did you do when you visited? And why did you choose to go to that particular small green space? This information will tell us how important small green spaces are to the sustainability of cities, but also help local councils make more informed decisions in designing and planning urban green space to ensure that the needs of people, as well as those of plants and animals, are being met. This will be crucial for designing our cities for the future. While 4.5 billion people currently live in cities around the world, this is projected to increase to about 7 billion by 2050, which will be nearly 70% of the world's population. It's crucial that our cities have lots of good green spaces so that everyone, no matter who they are, can have happy and healthy lives.
Thank you, Marie. Wonderful pitch. If it was up to me, my first question would be, what's your favourite urban green space? But I don't think I'm allowed to ask that because it's my job to pass on the questions from our audience. So I'm going to ask you that later. But the first question I have for you is whether there are differences in the benefits that people get from roof gardens or vertical gardens compared with traditional parks that we think of being on the ground. Have you looked at that at all?
Yeah, that's actually a really interesting question. There's been a lot of research on it, and it's something that I'm really interested in understanding is how different types of green space in cities, so whether that's green roofs or traditional parks or maybe even verges on the sides of the road, are they providing different benefits to people? And also, are they providing benefits to different types of people as well? So green roofs there's been a lot of research on green roofs and how they provide a lot of kind of stress relief and relaxation to people, particularly green roofs that are on office spaces. So, I'm sure many people have spent their lunches out on a green roof when they're working in the city. Whereas large parks are really important because they can provide multiple different benefits at the same time because they're larger. They can provide what we call like different zones. So you could have a playground in one place, a native garden in another place, and a large grassy patch for picnics in another location. So they have the capacity to provide a greater diversity of benefits, but we don't have space for these big parks. So while we'd all love to see lots of big parks in cities, we need to think more strategically about how to use these small patches and how to design them to get the benefits that people want.
Yeah, and it sounds like versatility, taking advantage of different opportunities to bring more green is really important. Great question next, and that is, what are your views on the importance of these green spaces as corridors for wildlife? So in urban areas, are these important for our wildlife?
Oh, 100%. Yeah. Increasingly, we're realising how important cities are for biodiversity. As cities get larger and larger, it's increasingly important that our cities are a refuge for biodiversity because they're running out of space in other locations. So we need to design our cities to be more biodiversity sensitive is what is often the word that people use. So designing small green patches or just green spaces in cities regardless that are connected is really, really important because they can create corridors for wildlife, particularly pollinators or even small mammals to be able to move, and birds as well. So that's a really good point, is that when we're thinking about where we want to put green space in cities and which spaces to prioritize, we really want to be prioritizing places that are going to increase that connectivity.
Yeah, for sure.
But that's definitely something that is really important.
And a really great question. Can people consider their own backyards a green space in the context of providing, services for people?
Yeah, 100%. So private backyards are actually a really important type of green space because while they provide benefits to the people living on those properties, like they can go out there, relax, exercise, they're also providing other benefits that everyone around them is benefiting from as well, such as kind of cooling down the area on hot days. It's not just the people living on that property that benefits from that. It's the people around it as well that are experiencing that cooling benefit. And things like helping flood mitigation, vegetation, the roots that vegetation has, the plants are really good at kind of preventing like sudden flooding, which is also really important. I guess the difference between private and public green spaces is mostly about who's able to access and benefit from them. And so we really want to, while private green spaces are really important, also really important for biodiversity as well, I should add. The public green spaces are of interest to me because there's something that everyone can access and everyone can benefit from. And so increasing public green spaces, we're kind of increasing that equality among people to be able to access that
exactly
space.
What I was thinking. Not everyone is fortunate enough to have their own green space in their own backyard. We've got time for one more question, and a question that I'm really interested in asking is, are trees like the most important? When we think of beautiful big gardens with lovely old trees, do you need to have trees providing ecosystem services for you to consider a green space kind of important or valuable, or is little bushes and shrubs and ground cover also equally important?
Yeah, I would say that every type of vegetation plays a role and can provide a benefit in some way. Trees, I admit, are often prioritized because they are really good for providing that cooling benefit. Like most other vegetation.
Shade.
Yeah, the shade, the canopy, really important. So trees are important, but that's not to say that other vegetation isn't doing anything. Shrubs are incredibly important for biodiversity. They provide habitat, food, particularly for pollinators. Grass is also really important to people because, one, grass can help cool down a bit, not to the extent that trees can. But grassy areas provide a lot of wellbeing benefits to people because we can picnic on them, we can relax, we exercise in large open grassy spaces. They're providing a huge amount of wellbeing benefits. Like we can't play soccer in a forest. It would be very difficult. So each type of vegetation plays an important role and provides a diversity of different types of benefits.
Well, thank you, Marie. Thank you for responding to our questions and for the fantastic work that you're doing. I'm picturing a soccer team becoming very good and directional with their dribbling if they were to play in a forest. But thank you, Marie. That was fantastic. Wow, what a fantastic start to our evening. Thank you to Rebecca and to Marie for kicking us off with such an excellent beginning to the night. And thanks to all of you for sending in such fantastic questions. I haven't been left hanging for even a moment wondering what you'd like me to ask. But before we hear from our next pitcher, what I'd like to do is introduce you to the people who I think definitely have the most difficult job tonight, and that is our wonderful judges. So you've already met Professor Moira O'Brien, Dean of the Faculty of Science. She is our head honcho judge this evening. Thank you, Moira. But Moira is also joined by her wonderful panel. So we have Belinda Smith, who is a cross-platform science journalist and editor. She's got wide experience in news, in photography, in radio, all sorts of wonderful things. She teaches, Belle does all sorts of incredible things. She's currently the online science reporter for the ABC, so you've probably seen her byline lots of times and heard her on the ABC. Next to Belinda, we have Phil Gardner, who's the Associate Director of External Communications here at the University of Melbourne. And in a former life, he was also a journalist and editor for News Limited and editor-in-chief for the Herald and Weekly Times. And he also consulted regularly on communications for the federal government. So welcome and thank you also, Phil. And finally, we have Dr. Laura Bibiana Ospino-Rosa, who is a research fellow in the School of Biosciences here at the University of Melbourne. And I would say she is our most empathic judge this evening because she competed in the big science pitch last year. And the reason she's here is because she won. So congratulations again, Laura, and we'll be hearing from you shortly. So thank you to all our judges for making the time to really think carefully about our pitches and to do the judging. And I'm sure the four of you have a lot of fun ahead, but also no doubt a very difficult task. So we're very grateful to you for being here. It is time then to move on to our third pitcher this evening, who is Dr. Felipe Martelli. Now, Felipe tells me that growing up, he wanted to be Batman. And then later, he almost failed his driver's license test because under pressure, he can't tell the difference between left and right. But I don't know whether both of those things have helped him, but they certainly haven't hindered him to now be based in the School of Biosciences and investigating insecticide resistance and the impact of climate change and insecticides, it would be helpful if I could say the word properly, on non-pest insects. So let's hear about Felipe's work.
When I was 4 years old, I was stung by a bee. This is one of my first memories, but somehow I don't remember the pain. All I remember is being mesmerized by the little creature. I think insects are amazing. 80% of all living things are insects. They recycle nutrients, are the backbone of food chains, and pollinate plants and food crops. But here's the problem. Around the world, insect populations are dying off. 40% of non-pest species, like beetles, butterflies, and bees, could disappear in the coming decades. That's a problem for ecosystems and agriculture. We don't fully understand how this is happening, but climate change and environmental contamination from the insecticides used in agriculture are two prime suspects. With this funding, I will work out how these two factors are killing non-pest insects and test ways to save key beneficial species. In my research with vinegar flies, I found something curious. Do you know how a metal chain left out in the weather rusts over time? Well, this process is called oxidation, and insecticides do something similar to non-pest insects, but from the inside out when they are exposed to small doses that contaminate environments. At these doses, insecticides wear out the molecules and cells in the insect brain, until, after many days, insects are out of action. With these results, I showed how insecticide contamination connects with the global declines of insects. That doesn't sound good, But here's the silver lining. I also found that if treated with antioxidants, the molecules that fight oxidation, flies exposed to insecticides survive. Now, because of climate change, I want to understand how insecticide contamination and high temperatures interact. I will examine the genetics and metabolism of flies and the native bees to answer why they are dying and test if antioxidants can save them. Look, insecticides are the top weapon against pests, and their use in agriculture keeps growing. Climate change is here, and it's affecting all of us. Understanding how they impact insects is essential to protect our ecosystems, our food production, and our future. I'm glad that bee stung me. It set me on the path that brought me here. And now, with your support, I can try to save them. Thank you.
So, Felipe, clearly getting stung as a small child was impactful, but you've also just told me that not only did you want to be Batman, but you also had the whole outfit with the cape. Was it wanting to fly that got you so interested in insects?
It might have been.
Okay, I'll ask you a proper question now, but I couldn't resist. First question is, what can people who like to garden and who value green spaces, like we've just been talking about, what can they do to protect insect populations? We all hear that insects are very important, but what can we practically do?
Well, in your garden, for instance, you can avoid using insecticides. There are some greener solutions. You can make solutions with water and vinegar, for instance, a low dose of vinegar, and that's sometimes enough to kill the pests in your orchids or around your garden. Things like the coffee powder after you drink like coffee, you can spray that, sprinkle that around pots, and that can also help preventing aphids and other insects that might be harmful to your plants. So there are all sorts of like good options there. And if you love bees, for instance, just have plants with purple flowers. Usually bees love them. So it's like there are all sorts of good options to do.
And purple flowers look gorgeous, let's face it. Next question from our fabulous audience. Could there be a molecular basis for the decline of insects globally, such as epigenetics from climate change? What a great meaty question.
Oh, wow. That's a fantastic question. Yeah, there could be. That's the whole point of trying to move on with this project, right? Because we really don't understand what are the proper mechanisms behind those declines. And understanding the mechanisms are important for a few reasons. The first one is because if we don't fully know the mechanism, we are blind to the full extent of what insecticides and climate are doing. And once we know the mechanisms, it's easier for us to, first of all, identify maybe signatures of stress to monitor wild populations and also test solutions. Epigenetics is definitely a factor playing there because things like temperature and the environment can affect how genes are going to be expressed. But what are the full extent of like epigenetics connecting with insecticides and climate? Yeah, this is something very cool to investigate.
Clearly your next postdoc. We know
definitely.
exactly what you need to do next.
Definitely.
Now we've got a fantastic question for you next, and this is from Rex, who is 9 years old. So I'm delighted that Rex is joining us, and welcome to all the other kids. I'm sure we have plenty of kids joining us this evening. But Rex would like to know which insect species is decreasing at the fastest rate. So which insect species is in the most trouble, I guess?
Oh, it's talking about species might be slightly complicated because it changes where you go on the planet. So we can talk in terms of groups.
Yeah.
So groups like bees, butterflies, beetles are usually the most affected ones. Other groups like flies, for instance, they might be actually increasing because they're occupying these vague niches that are being left, which is not good. Of course,
Who wants more flies?
They're definitely ecologically important and they have their relevance, but if they are occupying the space of beetles, butterflies, and bees, that starts to create ecological implications. And we never know how many species we can lose before the whole ecosystem collapse. So yeah, like which particular species, it will depend where you're coming from, but those three big groups, bees, butterflies, beetles, are the most affected.
Okay, excellent. And thank you to Rex for the great question. We've got time for one more question, and I happen to know that you are very good at demonstrating the honeybee waggle dance. I'm not going to ask you to do it now, but we do have a question whether insecticides impact that location, the waggle dance, the routine that tells their hive mates where food sources are. Is that dance being impacted by insecticides?
It definitely does, and there is a lot of research on this because what insecticides do, the majority of insecticides, they have targets in the insect central nervous system. And once the neurons are targeted, they can affect cognition. And the bee wagon dance requires so much communication between neurons, and that can really impact how well the bees can communicate. The wagon dance is a proper language with grammar and structure. And if the neurons are not communicating the way they should because of insecticides being present, they cannot give the proper message that they should be given to the other workers in the hive. So it is definitely affected.
Well, that's very depressing, but thank you for sharing it with us. I hate the thought of a bee doing this intricate, incredibly evolved dance and not being able to do it properly because of insecticides. So thank you for the work that you're doing, Felipe.
Pleasure.
Good luck tonight.
Thank you.
Wonderful pitch.
Thank you, Jen.
Now, a couple of reminders before we hear from our next pitcher. Please make sure you are thinking along the way about who you are going to vote for the People's Choice Award, because it's not so far away anymore and we can't wait to see who you vote for. And thanks so much to all of you for entering such great questions into our Q&A. Make sure you put them in as soon as you think of them. You're seeing that I'm not managing to get through all of them, so the quicker you get your question in, the better. I think it's time to hear from our next pitcher this evening, who is Dr. Oliver Eales. Now, Oliver moved to Australia from the UK 2 years ago, and he tells me that his first experience of Australian animals perhaps wasn't what us Australians might have dreamed of for him. It was when a possum climbed up his leg late at night in the middle of the city and completely terrified him and made him scream out loud. That was his welcome to Australian wildlife. Fortunately now, Oliver is based here in the Faculty of Science, and he's applying mathematical and statistical models to better understand the transmission dynamics of infectious diseases and inform public health responses. So take it away, Oliver.
If you've watched the news in the last couple of months, you've probably been hearing about a new strain of Mpox. But what's actually going on, and how could it affect Australia? That's what I'd like to use maths to find out. Mpox, or monkeypox as it was previously known, is an infectious disease that causes rashes, blisters, and fever. While most infected individuals recover after a few weeks, some infections can be severe, especially in children or pregnant women. Mpox can be split into two different types, which you've probably heard referred to as clades. These clades are simply named Clade 1 and Clade 2. Previously, both clades have only ever been found circulating in Central and Western Africa, where they were first detected in humans over 50 years ago. So you might be asking yourself, why have you only heard about Mpox in the last couple of years? Well, 2022 saw the first instance of widespread transmission outside of Africa. This global outbreak caused by Clade 2 of the virus made headlines when the World Health Organization declared it a public health emergency of international concern. The main mode of transmission of this outbreak was through sexual contacts, particularly between men who have sex with men. Now, this limited widespread community transmission and made the targeting of public health measures, such as vaccination to those at greatest risk of infection, far easier than if there were other modes of transmission. Fast forward 2 years, and the World Health Organization has just declared a new Mpox emergency, this time involving a new strain of Clade 1. Clade 1 is estimated to be more severe and is also predicted to have higher rates of non-sexual transmission, which may lead to more widespread and more complex transmission patterns. My project would use mathematical models to predict these patterns. A simple model might assume that everyone behaves identically, and that individuals are either susceptible to infection, infected with the virus and able to infect others, or immune to infection. Now we know that's not appropriate for Mpox, but we can develop more complex models that are. The model I'd develop would include differences in behaviour between age groups, different rates of sexual and non-sexual transmission, and also the effect of vaccination. The model can then be used to simulate what an outbreak might look like for certain values of the parameters that describe transmission. Now, using data from the 2022 outbreak, I would estimate parameters describing the past transmission of clade 2. Using what I learn, I would then explore future scenarios that might be expected when the new strain of clade 1 infiltrates Australia. For each scenario, I would develop optimal vaccination strategies that minimize the impact on public health and ultimately prevent deaths.
Amazing work, Ollie. Thank you. And I'm very glad it hasn't involved any more terrifying encounters with possums. The first question I have for you is, what are some other applications that might be appropriate for a mathematical model like yours? For example, are there other diseases? Are there other situations in the world that aren't diseases that this model could be used for?
So mathematical models have really been used for every disease you can think of. They're a really versatile tool for sort of assessing what's going on, what's causing transmission, and sort of applying interventions and figuring out what's best to do and when can we not do anything. And so it's been applied to respiratory viruses. You might have seen some stuff in the news about SARS-CoV-2 or COVID-19 back from the pandemic from a few years ago. There's been lots of work on influenza, especially during the 2009 pandemic. But there's also plenty of work on sort of other sexually transmitted infections like HIV and other diseases with completely different dynamics like mosquito-borne diseases like malaria or dengue. And even outside of human diseases, there's plenty of sort of applications for these sorts of mathematical models in animal diseases, in plant diseases even as well. So really there's nothing mathematical models can't really be applied to.
Which is why it's so important that we have people like you doing the work that you do. The next question I have for you is how your models are going to address the genetic differences of susceptible people in different parts of the world.
That's an interesting question.
Are there likely to be differences in susceptibility?
There are likely to be differences because there's been very different patterns in vaccination for smallpox. Now, smallpox is a different virus, but it has a sort of similar structure to Mpox, which means that people who were vaccinated against smallpox are likely going to have some immunity against Mpox infection. This is obviously going to be different across the world because smallpox vaccination campaigns have been very different. I think there's lots of people who have the smallpox scar on their arm, which shows they've got the vaccination, and it's basically it's somewhat older people who are more likely to have it, and younger people are less likely to have it. These rates are sort of available for most countries, and there's data on when smallpox vaccination was going on in Australia, so we can easily incorporate that into these sorts of models. The other thing which has something to do with immunity is how much of an outbreak they've had before with the previous smallpox.
Whether you've been exposed. Before,
So Australia had a very small 2022 outbreak, but they're currently actually in the middle of a much larger 2024 outbreak with the original clade 2 of the virus. So it's really unclear what's going on and whether there's some sort of vaccination effect or some sort of waning of vaccination effectiveness going on.
Interesting.
Or sort of,
Great question, and that is someone has said they assume that the virus strain is going to evolve every year or potentially, change quite quickly. Can the modelling that you're doing incorporate rapid changes, soon after they happen?
Yeah, so the modelling I would do would not be able to predict that sort of thing at all.
No.
But if there's some sort of evidence for a new change and some new virus emerges, some clade 3 or clade 1C or any new virus, as soon as it's sort of emerged, we can apply similar mathematical models and explore different scenarios, like what would happen if it's more transmissible or less transmissible, or if it spreads through different contact networks. That means that we can sort of assess the impact of a new strain emerging before one's even emerged. And then after one's emerged, we can sort of figure out what these new parameter values are and figure out what sort of scenario is most likely.
Interesting. Oh, there's so many good questions and we only have time for one more of them. How about this one? How contagious is Mpox and what kind of threat does it pose to the public?
So it's very unclear at the moment, but I would say that, we don't have to be worried too much about it at the current stage. There's evidence coming out of sort of Central Africa that it is more transmissible and that it might be more severe than the previous mpox.
Right.
But that's based on data which is in a very different context, and also based on data from the original clade, which has now evolved, and it's unclear if it completely applies to the new evolved pathogen. So really the answer is I don't know, and
Watch this space.
watch this space, yeah.
Yeah, well, thank you, Ollie. And I think it's really wonderful for everyone to hear about a topic that some people think, oh, maths, it doesn't apply to my life, to hear that actually it's essential for some really, really important work. So thank you for sharing it with us, and all the best for tonight.
Thank you.
So it's now my pleasure to invite Laura, who you saw earlier, a member of our judging panel this evening, who was the winner of last year's Big Science Pitch. And we've asked her just to come back and tell us a little bit about what the Big Science Pitch has meant to her. So thank you, Laura. We're so delighted that you were able to be here this evening.
Thanks, Jen. So have you ever wondered what's inside a beetle shell? I did last year during my Big Science Pitch. Some green Christmas beetles have a hidden white layer in their shells that acts like a parasol, shielding them from sunlight and still standing up to daily wear and tear. I wanted to examine beetle shells, hoping to unlock their unique properties because they could be applied to coatings to keep our cities cool. In the night of the event, I saw my ideas come to life through the questions from a really wide audience. It reminded me that my science is more than numbers, and it matters to my community, and it deserves to be shared. Standing under the big bright light, it was a little bit scary, but also exhilarating and empowering. And sometimes I meet people who remember my pitch, and knowing they care about my work is so heartwarming. While money can't buy happiness, it certainly can buy lots of microscopy tools. For example, a knife made out of diamonds or powdered gold. I need these to slice beetle shells in sections that are thinner than a human hair, and then image them with electrons. I also travel to visit museums and collect rare samples. Having a way to afford all of this is kind of buying happiness. But funding also makes priceless things possible. I can hire expert hands to enrich my project. I can train people to help me while being fair and paying for their time. And I have the privilege to test, fail, and try again, growing every time with every experiment. Because as the atomic physicist Niels Bohr said, an expert is a person who has made all the mistakes that can be made in a very narrow field. In conclusion, I'm incredibly grateful for the opportunity. It gave me perspective, funding, the confidence and freedom to push boundaries without fearing failure, and I could not wish better for tonight's excellent pitchers.
Thank you so much, Laura. And I think we couldn't wish for a better testimony to the power of the Big Science Pitch and why I love this event so much. So it's really wonderful to have you back and hear about some of the work that you've been able to do with your winnings from last year. So it is time to welcome our fifth pitcher to our stage. I can't believe we're already up to number 5. Our fifth pitcher this evening is Dr. Ludovica Monti. Now, from the School of Chemistry, Ludovica investigates the role of unique DNA structures to better understand how bacteria evolve and develop multi-drug resistance, which is a really pressing global health threat I'm sure we've all heard about. As well as doing that work, Ludovica tells me that she's brought her coffee maker with her everywhere that she's lived since her student college days in Rome. So whether she's been in the USA, the UK or Australia, she's always had the taste of home to kick off her day, and it's very important to her to be able to make a great cup of coffee while she's enjoying a new place. So hopefully she's also trying excellent Melbourne coffee. But let's hear from Ludovica now.
When I was a child, my grandmother would tell me fairy tales. And one story that stayed with me was the tale of Hansel and Gretel, who left a trail of white stones to find their way home through the dark forest. My grandmother's stories not only entertain me, but also instill a sense of curiosity about the world, which led me to travel to many countries with science as my guiding light and tackle key challenges like antimicrobial resistance. This fairy tale inspired my research. How can we trace the path through which bacteria develop resistance to drugs? Multi-drug resistant bacteria are superbugs that develop the ability to survive to antibiotic treatment, making infections much more difficult to treat and causing millions of deaths worldwide each year. If we don't take immediate action against antimicrobial resistance, this number will continue to rise. So how do we stop this? As a chemist, I study DNA, the molecule that carries the genetic information for life. Did you know that beyond the familiar double helix, which looks like a spiral staircase, DNAs can also fold into other fascinating structures? One of these unique structures are called G-quadruplexes or G-force, and they look like knots in a rope. These knots can be found everywhere in the bacterial genome, and they may help bacteria resist treatment. However, the path to developing drug resistance is still unclear. It looks like the dark forest Hansel and Gretel had to walk through. My goal is to use these G-force as white stones to trace the path that bacteria take towards drug resistance. First, we will create a map of G-force in the bacterial genome to visualize where these guiding stones are located. Next, we will expose the bacteria to various treatments and sequence their genome to track how these white stones change path as bacteria develop resistance. By understanding and following this path, we may be able to reverse it, and thereby resetting bacterial sensitivity to drugs and improving treatment outcomes. Uncovering how bacteria develop resistance will allow us to create better treatments that can save lives and reduce the burden of healthcare systems worldwide. Together, with your support, we can shine a light on the dark forest of drug resistance using G-force as our guiding stones to guide us safely home.
What a beautiful pitch, Ludovica. Thank you. And you've just told me that your grandma, who we've just seen, is watching you live now in Rome. That's very special. So I think we should all do a shout out to your grandma, who's clearly inspired you in many ways. And that leads me to ask, what inspired you as a chemist to start working on antimicrobial resistance?
That is an interesting question. So my background is in drug discovery and DNA biology, and I've my research has been primarily focused on parasitic diseases that affect mostly tropical countries. And I've been studying these unique structures of the DNA, which are called G-quadruplexes or G-force in these parasites. But I felt like that I could apply my knowledge and my expertise to also study other pathogens that cause this very critical problem globally. And this is what I aim to do with the researchers from this award.
Mm-hmm. Fantastic, thank you. A great question from the audience, and that is, what are your views on using bacteriophages that occur naturally in the environment to try and control drug-resistant bacteria?
That is a very good question. So there are many different approaches that now have been used to attack or to resolve antimicrobial resistance, and the use of bacteriophages is one of those. However, this goes a little bit beyond my knowledge, but I can say that there are perhaps some evidence or the possibility for bacteriophages to become resistant, or bacteria to become resistant to bacteriophages treatment. And so what we are proposing here is a new way of studying the disease in a way that then we are able to treat treat the disease and avoid the bacteria evolve towards becoming resistant.
Yeah, which presumably is the most important goal here. We've got a question of how do bacteria spread antibiotic resistance? I know about plasmid exchange, but are there other ways to spread immunity? What a great question.
Yes, this is also a very great question. So bacteria are very small microbes, and so there are many different ways that they can potentially spread these antimicrobial resistance. One of them is, for example, by exchanging genes that carry these antimicrobial information. And one of the things that we want to look at is the involvement or how G-force may be involved into driving this communication between bacteria and their involvement in regulating these AMR genes in a way that we could potentially be able to stop them.
Interesting. Let me see, there's lots of great questions. How about this one? Do you foresee bacteria gaining novel ways of resistance to your new treatments?
Thank you for this very interesting question. So, yes, what we are not proposing is to develop right now a new treatment. So, of course, because in this case we are talking about DNA structures, there is the possibility eventually that bacteria develop new ways to become resistant. What we want to do at this stage is to understand how bacteria goes from being a good bacteria to being a bad bacteria. And often we understand that the bacteria has become resistant at the end point when we treat the patient and we don't see any effect. But often the information that is in between gets lost. And so in a way we want to use these DNA structures or G quadruplexes as markers to understand how the bacteria evolves towards becoming a bad bacteria.
Fascinating. Well, thank you. I'm sorry we don't have time for more questions. I'm sure your grandmother is extremely proud of you, and thank you to any of your family members who've joined us from Rome. That's really special.
Thank you.
Well, it is time for us to welcome our very last pitcher to the stage. Amazing how quickly time flies when you're having fun. So let's now welcome Dr. Veronique Paris, who is from the School of Biosciences, and she's researching Australian vector mosquitoes by using advanced genomics and field experiments with the goal of reducing disease transmission. You can see we have some common themes here this evening. Now, a fun fact about Veronique, she tells me, her brother insists that everybody knows that she is fiercely competitive when it comes to Mario Kart. Apparently, years of intense sibling rivalry have made her an absolute master of the blue shell. Now, I've never played Mario Kart, so I don't even know what that means, but I'm sure she's excellent at it. So let's welcome Veronique and hear about her work.
We all can't wait for summer, right? To enjoy our backyards in the evening with a barbecue sizzling. Well, I hate to be the no fun person, but that peaceful evening might be putting you and your loved ones at risk. There's a good chance some uninvited guests will show up and they're not here to bring any food for the barbecue, but to make you their meal. I know I'm running the risk of sounding like someone you wouldn't invite to your next barbecue, but I'm talking about mosquitoes. When most of us think about mosquito-borne disease risk, we usually think about the big names like dengue or malaria, and we usually associate those with the tropics. But here in temperate parts of Australia, we're facing an increasing threat from locally transmitted diseases like Ross River, borreliosa, and Japanese encephalitis. These diseases can be hard to control, and that's partly because the mosquitoes spreading them haven't been well studied. Over the past few years, my research has focused on these lesser-known disease-spreading mozzies from our backyards. From advanced DNA sequencing techniques to ecological investigations, I'm on a mission to know our unwanted barbecue guests inside and out. One area that has recently caught my attention is insect-specific viruses. As the name suggests, these viruses only infect insects, not humans or other animals. So you might be wondering why I would be interested in them if they don't harm us. Well, until recently, these viruses were considered just part of the normal community of microorganisms living inside of the mosquitoes. But I think there's more to it. Some of these viruses might compete with pathogens inside of the mosquito, preventing transmission by blocking the pathways the pathogens need to be transmitted. This could help explain why some mosquitoes spread certain diseases while others don't. But even better, if I can identify which specific viruses do this, we might be able to harness their power and turn disease-spreading mozzies into harmless ones. My research will start by sequencing backyard mosquitoes from area that differ in their ability to transmit Ross River virus. By comparing these populations, I hope to uncover differences in the insect-specific viruses they carry. I will then characterize those specific viruses and find out what role they play within the mosquito and how we might be able to use them. But finding a way to stop disease transmission using insect-specific viruses is just the tip of the iceberg. On the way up, I hope to broaden our understanding of how mosquitoes acquire these diseases. From the environment, from organisms they feed on, and what functions do they serve? Can they help the insect adapt to the environment? I can't wait to answer at least some of those fascinating questions. With this research, I hope to learn more about life surrounding us, and most importantly, to make our backyard safer. And maybe then I'll finally be the fun person at backyards get-togethers without having to lecture about long sleeves and insect repellent.
I'm sure you are already the fun person at parties, so I think you're doing yourself a disservice. Well done, Veronique.
Thank you.
Amazing pitch. And I'm thinking you are not going to be surprised to know that the first question I have for you is probably one that pretty much everyone who knows you asks, and that is, what is your advice for stopping people being bitten by mosquitoes? Everyone in the room, put up your hand if you feel like you're the one who always gets bitten. What do we do?
Yes, that's indeed a very common question, and that's why at least I think other people think it's not a non-fun person answer to this. Obviously, wearing long sleeves and insect repellent, as I just said, is a good way, especially if you're in areas where you know that there's disease transmission happening. Other than that, if you have a backyard, there are a lot of different sort of easy things people can do. Make sure that there's no water holding bodies around your backyard. So if you have just a little pot plant with a saucer underneath, that can breed mosquitoes. And people get surprised by this all the time. I've done a lot of sort of backyard surveys where people tell me, well, I don't know where they're coming from. I don't have anything that breeds mosquitoes. And I find them very quickly because it really just needs like this little bit of a handful of water that can breed them. So make sure you don't have any standing water around your property is basically the easiest trick.
So my family has a dog who runs our entire family. Her name's Molly, and she loves to swim, and she has her own little pool in the garden. And I'm horrified by how quickly it gets full of, mozzie larvae, and we have to empty it out. So I'll take that advice.
The thing is, you can keep that, just tip it out every now and then, because they need a while to kind of emerge to be the biting adults. So if you just tip it out every now and then, or she, by swimming in it, then you will be safe.
Excellent. So we can make sure she can keep swimming.
Yes.
Thank you. Okay, next question from our audience is, can harmless viruses that you find in mosquitoes compete or inhibit harmful ones?
Yeah, so that's kind of the idea about this pitch, is that they might be able to sort of prevent viruses to be transmittable. And the way that most viruses get transmitted, so pathogenic viruses, is they need to replicate within the mosquito and then kind of travel through the body to the saliva glands and then get transmitted when they bite a person or an animal.
Okay.
And the way they do this, they kind of be sneaky ninjas and kind of use pathways that are already existing within the organism, within the mosquito. So I hope that we find viruses that are harmless, that are living in mosquitoes anyways, that kind of block that pathway already because they're using that for their own replication, but they don't cause diseases in animals or humans.
Yeah, okay. Interesting. Thank you. Okay, our next question is, what is your hunch about the relative importance of environment and blood meals as the dominant source of mosquito-only infections, and why?
Right. I don't know if I get the gist of that question correctly, but is it sort of what reservoirs there are in the environment that cause diseases?
So, yes.
That certainly is a very important point and is part of every sort of vector-borne disease research, because most diseases need like another host reservoir or something where they can sort of thrive to then be brought over to humans.
Yep.
But I would say that's very sort of virus specific. Some are very good at just going from human to human without any intermediate step. But it's definitely very important to study sort of animals and what they play in that sort of cycle of transmission for sure.
Interesting. Thank you. We've got time for one more question. The question is, are there particular qualities that make mosquitoes more or less likely to be carrying diseases? Obviously, there's different species of mosquitoes, but are there other things as well?
This is a very good question, and honestly is something that I we're really interested in ever since I started working on mosquitoes, is why are some very closely related species able to transmit diseases while the other one isn't. That's really something that always kind of struck me as a bit, why is that? That's kind of, I got really curious about this, and that's part of this research idea as well, because maybe some species carry those insect-specific viruses that kind of, make them not be able to transmit a pathogen, while the other species doesn't. And maybe that's, impacted by the environment they live in or just by the genetic differences between the species. But that's certainly something I'm still curious about, and I'm trying to get a bit more information about with that research.
Fantastic. Well, it's good that you're still feeling curious. I mean, it would be a very sad day, I think, if we stopped all feeling curious about what's going on around us.
Yeah.
Thank you, Veronique. That was fantastic, and thank you for answering our questions. So we are now at the end of our pitches, our presentations for the evening, and I'm sure you'll agree with me that the quality of tonight's pitches were just amazing. So a huge thank you to our 6 finalists, both for their amazing work and for the beautifully eloquent ways that they've shared their plans and their research plans with us this evening. What this means now, since we're at the end of our pitches, is that it's time for you to do some work. It is time for us to open the People's Choice Award. So please make sure you have your say and vote for your favourite pitcher now, because the winner of the People's Choice Award will receive an additional $10,000 to put towards the work that they have just been pitching to you. And I'm sure you'll agree that there's really extraordinarily important and useful work that has been pitched tonight. So please follow the link in the chat now so that you can register your vote. All right, what needs to happen now is that we need to give our judges a few minutes to confer, and so that means that it's time for me to test you on how well you've been listening. Now, I know I didn't tell you that there was going to be a test at the end, but you've just discovered that there is. So our online team is going to hopefully share some trivia questions with you, which are based on each of tonight's pitches. So we asked each of our pitchers to give us one question, a tricky question to test you on how well you are listening. So get your thinking caps on, and once we share the question, you can vote for your answer. So let's see if we can get our first question up, which is a question from Rebecca. How many frog equivalents can Rebecca fit onto a cell culture plate? So she did actually tell us a specific number. Whether you can remember it or not is going to be the next test. So your options here are the equivalent of 24 frogs, 48 frogs, 72 frogs, or 96 frogs. And I can see we have lots of answers in. So I think we can probably jump and show you what the response was. And 68% of you remembered correctly that the right answer is 96 frogs. So well done for everyone for listening to Rebecca so carefully. All right, our second question is from Marie. So remember, talking about urban green spaces. And Marie's question for you is, which of the following is an ecosystem service that an urban park might provide to urban residents. So that is all of us. And the options she's given you are food to eat. Oh, I'm a very big fan of urban gardens with lots of nice veggies planted. Temperature reductions on hot days, flood control, or that always sneaky possible response in multiple choice questions, which is all of the above. And I can see we have lots of answers in, and you guys have yet again done an excellent job. You are right that it is all of the above. I think that was meant to be a trick question and none of you got tricked, so well done. Okay, I think this is my favourite question. We have a question from Felipe, and you would have had to pay very close attention to get this one. Felipe would like to know whether you listened and whether you remember how old he was when the bee stung him. And I want you to picture that as well as the bee stinging him, he was also jumping around in his Batman costume. So the questions or the possible answers you have are, was he 3 years old? Was he 4 years old? Was he 5 years old, or was he 6 years old? And I think the answer in every case was he was probably really cute in his Batman costume. Yet again, you are doing very well. I can see the answers as they are coming in. And you have all guessed correctly that Felipe was 4 years old when the bee stung him. Well, I guess you weren't guessing because you remembered because you paid such good attention. All right, let's move on to our next question. This is a question from Oliver, and Oliver's question is, which continent was Mpox first detected on over 50 years ago? And your options here for the continents are Africa, South America, Asia, or Europe. No Australian option for us, and I'm guessing we probably all remembered what Ollie told us about Mpox in Australia. So Africa, South America, Asia and Europe. And you will recall, as 94% of you have remembered, that Africa is the correct answer. That is where Mpox was first detected more than 50 years ago. All right, let's get on to our fifth question, which Ludovica has given us. And her question is, what will allow us to discover new mechanisms of antimicrobial resistance? Your options here are pebble stones, which I just think is a fantastic answer given how much I'm sure we all loved Ludovica sharing her childhood connection with Hansel and Gretel. Your next option is secondary DNA structures known as G4s. Hansel and Gretel, surely they would be helpful in this situation, or the dark forest. And I think Ludovica is teasing us a little bit because I'm sure that you all remember that, of course, it wasn't Hansel and Gretel or the pebble stones or the forest. It was these secondary DNA structures that she told us all about and we all learnt about this evening. So thank you, Ludovica, and well done to everybody. All right, moving on to the pitch that we just heard from Veronique about probably one of our least favourite things, mosquitoes. And Veronique's question is, why are insect-specific viruses interesting for mosquito-borne disease research? The options are, firstly, that they infect humans and mosquitoes easily. Secondly, that they may block the transmission of diseases within mosquitoes. Thirdly, that mosquitoes need them to transmit diseases like Ross River virus. And our final option is, and remember, Veronique told us she wants to be the fun person. The final option is that they make mosquitoes tell better jokes. I think she's having a lend of us. So the answer, of course, which most of you have remembered, is that they may block the transmission of diseases within mosquitoes. So I am really impressed. I know that some of them were easy questions, but you know, people are tired, people are busy. The fact that you've all listened and recalled pretty much all of those, I think is really fantastic. And you know, this is part of the point of the Big Science Pitch. Yes, it's to celebrate our wonderful researchers, but also for us to all learn some really interesting current research that's going on. So Hope you had fun with our little trivia game. But we have dilly-dallied for long enough, and I know that our judges are in fact ready. They've made their decision and they are ready to announce our top 3 pitches. So I'm going to hand back to Professor Moira O'Brien and ask her to tell us a little bit more about the judging that happened this evening, about the process, and then we're going to hear from our judges about who our winners are for this year's Big Science Pitch. Thank you, Moira.
It is indeed announcement time. Using a scoring matrix, we judges have assessed each pitch against several criteria, which are as follows. Was the pitch innovative? Does the pitch idea feature innovations which are likely to make headway in the particular field of research? Was the pitcher engaging, clear and enthusiastic, explaining the research idea in a way that is accessible to a broad general audience? Could this research possibly support real change in understanding methods, theory or their application? Now remember, we are looking for real world benefits, positive social, economic and environmental outcomes. It is a lot to ask, but as we face unprecedented global challenges for the sake of our future generations, the Faculty of Science has ambitious goals for our research, as do our staff and students. We are working with communities, industries and governments to address the crises of climate change and biodiversity loss. We are designing technologies and using data wisely to enhance human lives, nurture the natural environment and increase social justice and resilience. We are developing food systems, therapies and pharmaceuticals of the future, helping to nourish the world and improve the health and welfare of all living things. And if you want to learn more about this critical work, I encourage you to explore the faculty's website, read our researchers' articles on the university's most popular pursuit platform, or attend our free public lectures throughout the year. But now I'd like to hand over to Phil Gardner to announce the 3rd prize winner.
Thank you, Moira. And without further ado, I'd like to announce Veronique Paris as the winner of the 3rd prize for her work on reclaiming our backyards, new avenues to sustainable mosquito control.
And I get the fabulous job of announcing the 2nd prize winner, and that tonight is Felipe Martelli for his work on protecting non-pest insects from climate change and pesticides. Well done, Felipe.
And I get to announce the big prize. And I am absolutely delighted to say that the winner of the 1st prize is Rebecca Webb. Well, I'm sure that everybody involved in this event is feeling quite exhausted by their efforts. I've no doubt the audience is feeling elated though. My heartiest congratulations to all of our finalists for their excellent pitches. Thank you to my fellow judges, Belinda, Phil and Lara, for applying their expertise to separate this close-fought competition, and indeed it was extremely close. I am particularly grateful for your expertise and your time, so thank you. Huge congratulations to our winners. I am absolutely thrilled for you. But wait, there is more, and I'm going to now hand back to Jen to announce the winners of the Tim Winton's Choice Award and the People's Choice Award.
Thank you, Moira, and congratulations already to the winners who've been announced. And I'm so glad Rebecca had a good reason to be awake most of the night to join us from the US. But as Moira said, I do have the great pleasure of announcing the winner of the Tim Winton Award. As I said, Tim can't be here. He's just been on a huge tour for his new book. And I must admit, I was extremely excited and delighted when he actually rang me yesterday. I answered my phone and it said, "Hi, Jen, this is Tim Winton." And I thought it was maybe a prank call, but it wasn't. He was calling me to share his choice this year. And this is what he asked me to share with you this evening. So Tim says, "As with every year of the Big Science Pitch, these pitches are all excellent and worthy. I'm excited by how much good work is being done by young scholars in this space, and I'm very happy to be associated with the Big Science Pitch. It's been very hard this year to make a call about which one to support, but because we're facing massive ecological losses under climate change, and the brunt of those losses will be borne by insects, which are already besieged by surging inputs of insecticides, I think Felipe's pitch is the most compelling, and I'd be proud to have his work given the award in my name." So congratulations to Felipe, winner of this year's Tim Winton Choice Award. I have one last exciting task this evening, and that is to announce the People's Choice Award. I think this is our most exciting award. The whole idea of the Big Science Pitch is for us to share our research with people all over the place, people who are into science, maybe people who aren't into science. And I think it's fantastic that so many of you have joined us this evening and voted. Of course, it was a close race this year, it always is. But there's a bit of a theme going here. You perhaps won't be surprised to hear that the winner of the 2024 Big Science Pitch People's Choice Award is, drumroll, Felipe. Woohoo! What an amazing outcome for Felipe. That's a lot of research funding to help him do what I'm sure you'll agree is really very important work. So I'm delighted. And I'm delighted really for all of our pitches tonight. It's not easy to explain complex work to a broad audience in 3 minutes, to come into a studio when you might have not ever spoken to a camera before. You know, this is not easy, and I'm incredibly proud of all of our pitches, and I think they've all done themselves incredibly proud. So well done. I have the joy of now making a few thank yous. Thank you, of course, to our judging panel, Moira and Belinda and Phil and Laura. They put a lot of time and thought into making their decisions, and we're very grateful for their time and their expertise. Laura's saying it was really hard. I would like to say thank you to the Science Faculty event team and all of the talented crew in the studio here, who I can see but you can't, who've done such a lot of work to help tonight come together in such a successful way. Special thank you to Taylor and Holly and Susan and Lee and his amazing team. I won't name you all, but we're very grateful to all of you. As I've said, thank you again to our pitchers, to Beck and Marie and Felipe and Oliver, Ludovica and Veronique for the time and effort they put into their pitches this evening. A huge thank you to all of the generous supporters of the Native Australian Animals Trust for providing some of tonight's prize money and for supporting our incredible up-and-coming research superstars in a really meaningful way. Research funding makes a huge difference. And finally, thanks to all of you for joining us online, supporting our pitches, asking great questions, registering your votes, playing along with our silly little trivia quiz. Thank you for all of the ways you've contributed, and particularly for deciding on the People's Choice Award. So that is it from us. Congratulations to everyone involved, and thank you for your involvement. And we really look forward to seeing you again next year for the Big Science Pitch 2025. Have a great evening, and thanks again.